Hello everyone, my name is Renata Pavlovic and I will talk today with you about my favorite dessert. It's ice cream. And I will give you some insights on how we can get the flavor into ice cream and how we can make this more sustainable. When I started preparing for this talk, I had to think of all these slogans. One scoop at a time, or more flavor, more fun, a world of great taste. I had to think of childhood memories and when I went to get ice cream, what I was feeling, and at some point in my time as a researcher, I had to think of where does this flavor come from? And today I will tell you how we get flavors conventionally, and then I will show you how we can use microorganisms to obtain these flavors. And in the end, I will ask you if all our work as research scientists is worth the effort. Ice cream has no inventor. We know that in China there was a similar product to ice cream many years back, where they combined fruit juice with snow and called it ice cream. So this knowledge spread to ancient Greece and Rome, where they had similar approaches. Then we lose track of ice cream in history. We don't know what happens, and only in Italy in the around 1200, when Marco Polo returned from his journeys in Asia back to Europe, he brought that knowledge back and the ice cream spread through Europe. And in the 17th century, we had ice cream almost everywhere, but only at royal courts. So it was a luxury product. And by the end of the 19th century, we have ice cream production on an industrial scale. So it's available to many people because we have refrigerators. So I want to share how people favor ice cream flavors. We have in the US, those are numbers for the US, but when you look at other countries, you get a similar pattern. We have vanilla, which makes up to 30% of all the ice cream that we consume. On second, third, and fourth place, we have chocolate. We have cookie and cake or chocolate chip flavored ice creams. And there is even an ice cream shop in Venezuela that claims to have 860 different flavors. So the flavor is basically only limited by our imagination. 860. I don't believe it, I haven't seen it. If maybe somebody of you went to Venezuela, please tell me if that's true. Where do we get this flavor? Where do we get vanilla? Where do we get chocolate, strawberry, banana? I'm a big fan of peach ice cream, by the way. So let's take a look at how ice cream looks like. What is in there? What do we have? What makes the flavor? So for once, we have air cells. Those are the white areas here that you can see. My laser pointer doesn't work. I didn't know that. So the white areas inside there are air cells. And if you have small air bubbles or air cells, then you will have a very creamy ice cream. But if those air cells are very big, then it will be very fluffy. Air can make up to 50% of your whole ice cream volume. So if you pay for ice cream, you pay 50% for air. We have ice crystals that are also important. If you want to make ice cream creamy, you will use smaller ice crystals. If you want it, let's say, more crunchy, not that creamy, then you'll use larger ice crystals. Now the third most common ingredients are fat droplets. Fat droplets in this case contain glycerol and a fatty acid. The fatty acid that you see here, it's palmitic acid. It's not very healthy. Most likely you've heard of omega-3, omega-6 fatty acids, the healthy ones. You should eat fish and so on. So this one is not the healthy one. It has a straight chain, so it's not very healthy, but we still need it because it will give our ice cream the shape that it needs, and it will also be part of the flavor that we taste. The liquid syrup is the last most common ingredient, and that's where the sugar is. We have sugar molecules that give us the sweet taste of ice cream. Different quantities, depends on your preferences. But the most important molecule that I want to show you there is the vanillin. That's our flavor molecule. And that flavor molecule can be exchanged for different molecules without changing any other ingredient, and you'll get a completely different ice cream. If I want strawberry ice cream, I'll put a Forel there. If I want peach ice cream, I'll just exchange the vanillin for a gamma-decalactone. But all these flavors need to come from somewhere. And we get these flavors, or these flavor molecules, we get them from plants, usually from plants. I want to give you an example on how this procedure works for vanillin, and it's comparable to many, many other flavor compounds. We start with a plant. In this case, we have for vanillin, Vanilla planifolia. It's an orchid. It's very picky, 26 to 29 degrees through all the time that it grows. It needs special bees to pollinate those flowers. And once they're pollinated, they will form very long pods. You can see some of them already here forming. So once they form these pods, and that takes 6 to 9 months for them to grow to approximately 30 centimeters, long pods. Once that happens, they can be picked. After they're picked, we need, again, 3 to 6 months to dry them and to prepare them for the actual consumer. This is how they look like. We have pods that are dried, that have now a lot of vanillin, the molecule that I mentioned in the ice cream, and we pay a lot. We pay a lot of money to get 1 kilogram of vanilla pods. Starting from the plant, we need 6 kilograms of green pods to get 1 kilogram of dried vanilla pods. I don't know if you mind paying $500 per kg for something that you eat very frequently. The price is also very dependent on how the harvest was. How did the plants grow that year? That's why it's a range. Last year, the harvest was very bad. The quality of the pods was not good, so you had to pay $600. This year, it was not too bad, so we have $500 paying for 1 kg. Madagascar is one of the main producers. They have 60% of all the vanilla that you could get. It's produced in Madagascar. But as you've maybe recognized, the whole process from plant to the end product takes a lot of time. You need to invest a lot of money, and that will also reflect in the price. And if you, for example, switch to cocoa bean, which has a much faster turnover rate and is not that picky, then you can make more money with less effort, and that's what Madagascar is also doing. So we have many challenges that we face when we think of plants, using plants to get our flavor molecules. One of them is the availability. As I mentioned, we need special bees, at least for the vanilla orchid, that can pollinate those yellow flowers. Then it's also climate dependent. 26 to 29 degrees, long stretches of sunshine, and it's very specific growth moments. They need a lot of water, a lot of rain. And not only this, but also the use of fertilizers in some stages is necessary, because the land that they're growing at is not that good. Which brings me to the point of sustainability. The One North Festival is a lot about sustainability this year, and if we think about Brazil in the recent months, or if Indonesia in the last few years, or maybe even today, if you look outside, if there would be a window, we know that rainforests are burning. And we know some of them are by accident, by nature, it happens. But some of them, and I dare to say most of them, are due to human hand. And it's to get more arable land. So we burn them down to get more arable land. We will plant our cocoa beans or citrus fruits or palm oil, palms. But after some time, this land won't be good enough. So we need to use fertilizers and so on. So it's not a good cycle. And taking all these three together, we face quality issues. We can't make sure that the quality of the products that we want will be the same every year. It's not only vanilla. It's many different flavors. As I mentioned, we have strawberry, we have banana, and so on. And they all have a certain chemical or a certain molecule, a certain flavor molecule that you can extract, and that they will give you the certain flavor in your ice cream. So thinking of strawberry ice cream, it's the one up there, the furanol. We use that one in ice cream and we will get strawberry ice cream. We'll get citrus ice cream. The idea showing you this is that these molecules are the ones that are responsible for the taste of our ice cream and other food products. And we don't need necessarily to use those fruits. I want to focus my talk on these two, on decalactone and gamma decalactone and gamma dodecalactone. Gamma decalactone smells like peach, and gamma dodecalactone is a combination of coconut and peach. And I want to show you how we can use microorganisms to produce these flavor compounds. When I say microorganisms, we at Biotrans, well, it's the short form for Biotransformation Innovation Platform. We at Biotrans work with different microorganisms, and we can choose from many of them. We have 1,500 yeast species that we could cultivate in the lab and that we can grow in the lab. We have 30,000 bacteria that we could work with, and over 120,000 fungal species. So those numbers only tell you how many we can work with in the lab. But there are over estimation between 2 and 3 million more fungus out there that we're just not able to cultivate. So we're lacking techniques to be able to use that variety in nature. So let's focus on those that we can handle. What do we do with them? We can, in order to get large quantities of these organisms and subsequently the flavor compound that we want, we use these fermentation vessels. For those of you who like to cook, it's basically a pot. In this fermentation vessel, we can control the temperature, we can control the pH, we can control the oxygen, we can control the ingredients that our microorganisms will get to grow. And once we have these controlled conditions and we have very happy bacteria that grow and produce, we can get our flavor compounds. And when we start what we say low value feedstock, so something that is of low value, which would be glycerol, wastewaters, or food waste. So basically or plant materials, plant waste materials, we can use those to produce our flavor compounds. We face challenges. It sounds too easy. It sounds too easy to just do that. And to make it more efficient, we need techniques to get organisms that are more efficient, that can produce more of our flavor compound. What we do is we can learn from nature. We can learn from the plants. We can look at the plants, how they produce the flavor compound, and then we can transfer that knowledge to microorganisms that we can manipulate, genetically manipulate. What does that mean? Or just very rough, what does that mean? We choose a microorganism that we like, that we can manipulate, and we look in nature. In this case, we look at peach, and we look how can they produce that flavor compound? How can they make this flavor compound? And we know the genes that are responsible for this, or the proteins that have this ability to make that flavor compound. And we integrate that, we combine the cells and that gene and force our cells to take up that gene. So once we have the cells with that gene, we can start producing. Now that would, again, be too easy. There are some aspects that we need to think of. So if we have the gene from the peach, we can produce decalactone. But that decalactone shouldn't kill our cells. So if it's toxic, then we need to think of maybe different cells. Or if your flavor molecule is volatile, what do? You come to the lab one morning, you grew your cells overnight, and you think, oh wow, our lab smells really nice like peach. Well, that's not what we want. We want it to keep it inside, right? We don't want to lose it. Because our flavor-producing cells should produce flavors that are kept in the solution and not in our lab. And also, we need to make sure that we give all the ingredients and all growth materials that our cells need during this process. There is another option. For people who don't like metabolic engineering, which is the official term of what I've just shown you. If people don't like that, well, we have another option. We do evolution in the lab. What we do is we start what I call original cells. So those are maybe cells that we found in nature, but they produce not a lot of the flavor compound. And we start with these cells and we transfer them to the so-called first stage. In the first stage, we will have a pressure on these cells to produce a lot of gamma decalactone, for example. And we will only take the top 10% of these cells and transfer them to the second stage. In the second stage, we will increase the pressure. We will only select strains that can grow fast and produce. Next stage. Again, we increase the pressure. You can compare that to finding the fastest and smart. In school, it would be, I start with a class of students and the first test is super easy. And all the A and A plus students get to the second stage. In the second stage, I introduce a new factor, and that is time. So it's easy questions, but you have to be fast. Only the A and A plus students get to the next stage. In this stage, it's time and more difficult questions. And so we increase the pressure with every step, and you can do that 50, 100 times. And in the end, you will have evolved cells. Evolution. We selected for the fittest. We selected for the strain that produces the best, that produces the highest amount, but that can also grow very well. That similar approach we took with production of gamma-decalactone. We have a sugar solution. We have an engineered baker's yeast, which means that we added genes from plants. We also removed genes from this organism to not make it degrade our product. We add sugar because it needs sugar to grow. And we transfer it, fermentation, to a small fermentation vessel. Now, the first one that I showed you was already 200 liters. We're working with a 1 liter. Let's start slow. And when we're growing those cells for 3 days, we need to separate our cells from the sugar solution from our produced gamma-decalactone. Downstream recovery, fancy word. What it means is that we need to get a pure product. We want to separate the things that we don't need. At this stage, we can produce 80 milligram per liter. It's not a lot. When you think of vanillin production, which is already in industrial scale, they produce between 11 and 15 gram per liter using bacteria. So they're pretty good. We're at the beginning. But that doesn't mean that our product can't be used as a flavor in an ice cream, for example. This whole process is very important, and we do it for every flavor compound that we're looking at. We start with discovery. We look if in nature we have strains that already produce the flavor compound. If they do, we go either for evolution or we go for growth, and then for separation, and then we have our product. If we can't find something in nature, in our microorganisms, then we learn from plants, go for pathway optimization, and then go through all the other steps. Science takes time. The stage that we are now at is a proof of principle strain, which means we could show that we can produce our flavor compound. We're here, but we want to get up there. For that, we need to increase the yield, titer, and productivity of our strains. It may take some time. I think 3 to 6 years is very optimistic. I would put it 10 years there. Especially if you look for existing technologies. If we only work on those technologies that I showed you right now, then it will take a long time. But if we at the same time work together on novel technologies, and maybe some of you have also heard about CRISPR-Cas, so these technologies, they help us to speed up the whole process. They help us to speed up the pathway optimization process. So in terms of strain development, it can make things faster. It's not only vanillin that is produced with microorganisms. We already have many products that are on the market that we don't know of. Well, I didn't know. Many of them are already produced with microorganisms. One of them, citric acid, Aspergillus niger, fungus, very fluffy, blackish fungus. All the citric acid, I don't know the direct percentage, but I'm pretty sure it's more than 50% of all the citric acid that you find in your food is produced by this fungus. It's a very good producer. Or xanthan gum, for those of you who go gluten-free, well, you've heard of it. You use that. It's produced by Xanthomonas campestris. It looks yellowish, glibbery. For me, a little bit strange. Okay, it's a bacterium. And then my favorite. Okay, not ethanol, but Saccharomyces cerevisiae. Used for wine, beer making, even if you're a passionate cook and you bake a lot, you'll use that. It's baker's yeast. And we use ethanol in many beverages, and it's now in discussion to use it as a fuel. It looks much nicer compared to campestris. It's a very nice white and shiny yeast. So we have three categories. We have fungus, we have bacteria, we have yeast, and they're already in there. They're out there in the industry, and they're being used. And the question is, why can't we do that with food flavors as well? We can. We can produce all these flavors using different microorganisms. We just need to consider three things. Smart people did that. They say you need to consider people, planet, and profit. And if all these three factors combined work together, then we can have a bioeconomy where all three factors are happy. People will get quality products. The planet will have a sustainable production of these, so we will save or at least protect the planet as far as we can. And the profit for the companies will also be there. We have many problems that we face. And I know that many people are doing many things at the moment to stop that and to prevent that. But we also know that it can be just a short-lived outrage, and that people will forget if we just wait long enough. And I just hope that you won't forget everything that you've heard today in this talk, and that you know that you will take home how to use microorganisms to produce food flavors. Thank you.